Cloning is the production of genetically identical copies of DNA, cells, or organisms through some asexual means. Gene cloning is the cloning of DNA to produce many identical copies of the same gene. Gene cloning can be used to identify differences in the base sequence between a normal gene and a gene that is mutated. It can also be used to genetically modify organisms in a beneficial way. Transgenic organisms are organisms whose genome has been altered by the insertion of a gene from a different species. They are frequently used to produce a product desired by humans. When cloned genes are used to modify a human, it is called gene therapy. Recombinant DNA takes DNA from 2 or more different sources. To make rDNA, a technician needs a vector to transfer foreign genetic material into a cell. Plasmids are one common vector. They are small accessory rings of DNA found in bacteria, and they can replicate on their own because they are not a main part of the bacterial chromosome. To introduce foreign DNA into vector DNA, 2 enzymes are needed: restriction enzymes, which cleave DNA, and DNA ligase, which seals DNA. The restriction enzyme first cleaves DNA at a specific site, creating a gap. Foreign DNA with complementary bases to the exposed ends of the vector are inserted. DNA ligase seals together the human gene in the plasmid, and the host takes up the recombined plasmid. Gene cloning occurs as the plasmid replicates. Complementary DNA is a DNA copy of human mRNA that is synthesized by the enzyme reverse transcriptase. cDNA is transcribed and translated by bacteria to produce a human protein. This can happen because the genetic code is the same in humans as in bacteria.
The polymerase chain reaction, or PCR, was developed by Kary Mullis in 1985. It is used in research labs to create copies of a segment of DNA quickly in a test tube. The PCR mimics DNA replication in the cell, except for the fact that it is specific. It amplifies a targeted DNA sequence and requires DNA polymerase and a supply of nucleotides for the new DNA strands. It is called a chain reaction because the targeted DNA is repeatedly replicated as long as the process continues. The PCR can be analyzed for several reasons. An example is mitochondrial DNA taken from modern living populations, which was used to decipher the evolutionary history of modern populations. Additionally, DNA is taken from a corpse burned beyond recognition, and it can be matched to the bristles of that person's toothbrush using the PCR. Other uses of the PCR are DNA identification, finding relatives, confirmation of viral infections, genetic disorders or cancers, and crime investigations. Also, fingerprinting through short tandem repeat, or STR, profiling has helped identify victims of natural disasters. Gel electrophoresis is a process of analyzing DNA following the polymerase chain reaction that separates DNA fragments according to their size, which results in a pattern of distinctive bands that identifies the person. Short tandem repeat, or STR, profiling has been the method of choice. STRs are short sequences of DNA bases that recur several times. Using STR profiling is advantageous because it doesn't require restriction enzymes. Since the chromosomal locations for STRs are known, it is possible to subject only these locations to the PCR and use gel electrophoresis to arrive at a band pattern that is different for each person. The newest method that has been developed for producing DNA profiles, called fingerprints, doesn't use gel electrophoresis. Instead, DNA fragments are fluorescently labeled. A laser excites the fluorescent STRs, and a detector records the amount of emission for each DNA fragment.
Genetically modified organisms, or GMOs, are organisms whose genetic material has been altered or enhanced using DNA technology. The products of GMOs are called biotechnology products. Products of genetically modified bacteria include insulin, clotting factor 8, human growth hormone, tissue plasminogen activator or TPA, and the hepatitis B vaccine. Uses of transgenic bacteria include the promotion of plant health, the cleanup of beaches by bacteria which eats oil, and the removal of sulfur from coal before it is burned. Techniques have been developed to produce genetically modified plants as well. In one, protoplasts, which are plant cells without cell walls, are treated with an electric current and suspended in a liquid containing foreign DNA. The electric current creates tiny holes in the plasma membrane, which allows DNA to enter the cell. These protoplasts can later develop into mature plants. Foreign genes inserted into plants can be used to create resistance to pests and herbicides. Transgenic plants can also be engineered to produce certain human proteins in their seeds. Gene farming is the use of transgenic farm animals to produce pharmaceuticals. Genes that code for therapeutic and diagnostic proteins can be incorporated into an animal's DNA. The proteins will later appear in the animal's milk. To produce transgenic mammals, DNA containing the gene of interest is first injected into donor eggs. In vitro fertilization takes place, and zygotes are placed into the host females to develop. After female offspring mature, the product is secreted in milk. Unfortunately, animal cloning is a very difficult process with a low success rate, and the majority of cloning attempts are unsuccessful.
For many years, researchers believed adult vertebrate animals couldn't be cloned because cloning requires all genes of adult cells to be turned on for normal development. In 1997, however, Scottish scientists announced that they had cloned a sheep which they called Dolly. Since then, many other animals have been cloned. In order for transgenic mammals to produce a product, enucleated eggs from the donor are micro-injected with 2 N nuclei from single transgenic animals. Then they are coaxed to begin development in vitro. Development continues in host females until the clones are born, in which female clones have the same product in their milk as donors have in their eggs. Transgenic mice have been used for many research products. An example is a section of DNA called SRY, which is the sex-determining region of the Y chromosome, which produces male animals. The SRY gene was cloned, in which one copy was injected into single-celled mouse embryos. The injected embryos developed into males, and the embryos without the injection developed into females. Genes can also be eliminated. A knockout mouse has been used in which both alleles of a gene are removed or made non-functional. An example of this process is a knockout mouse lacking the CFTR gene, which is the same gene mutated in cystic fibrosis patients. The phenotype in the mouse is similar to humans with cystic fibrosis and can be used to test new drugs for treatment of the disease.